Hi everyone, welcome to my A-level biology lecture series. In this lesson, we will explore codominance. At the end of this lesson, you will be able to interpret and construct genetic diagram, including Punnett square, to explain and predict the results of the monohybrid crosses that involve codominance. Let's get started. Codominance is a form of inheritance with both allele in a heterozygous genotype are fully and equally expressed in the phenotype without one being dominant over the other. For example, the HBA allele code for normal beta polypeptide and HBS allele, which code for the defective beta polypeptide, are codominant. So you can see that in this table, the normal allele and the sickle cell allele, both of them code for the same polypeptide, which is the beta globin. But due to the single base substitution, so therefore these two allele are given different symbol. So for normal beta polypeptide allele, it is given HBA allele, and for the sickle cells allele, we give them this. We give this allele with the symbol HBS, and both allele they are codominant. So in carrier, which the individual with a heterozygous genotype, HBA and HBS, be careful, the heterozygous here, we call them as carrier, okay? Both normal and abnormal hemoglobins are produced. So therefore, this is a special phenotype. Why? Because we can detect the normal and as well as the presence of abnormal hemoglobin are produced. So when both parents are heterozygous carrier for sickle cells, in this case, with genotype HBA, HBS, there is a 25% probability that their child will inherit the HBS allele from each parent, resulting in the homozygous HBS genotype. So when two parents, male parent and female parent, both of them, they are carrier, with the genotype heterozygous HB and HBS, their gametes will be two different kinds of gamete. 50% of the gametes with HBA allele and another 50% with HBS allele. Let us look at how and what are the probability of the combinations when these two different type of gametes fuse together. So from this Punnett square, we can see that 25% probability of each child will be normal, and 50% will be carrier, and the remaining 25% will be sickle cell anemia with two copies of HBS allele. So from here, we can see that the phenotypic ratio, normal to carrier to sickle cell anemia, in the ratio of 1 to 2 to 1. So we wouldn't be able to get back the Mendelian's typical ratio of 3 to 1 in this case, because the carrier is a new phenotype. So in this example, the typical 3 to 1 phenotypic ratio seen in a simple dominant recessive inheritance is not observed. The HBA and HBS allele are codominant, so both are expressed in heterozygous carrier. So this results in the third distinct phenotype, and we call it as a carrier, which would not appear in the dominant recessive cross. The heterozygous individual express both parental phenotypes seen in the homozygous condition. What is this? The meaning of this? Because HBA individual produce only normal hemoglobin, HBS individual produce only sickle hemoglobin, and HBS heterozygous or heterozygotes produce both normal and sickle hemoglobin, showing characteristic of both homozygous parents. Another example of codominant is observed in the camellia flower color, When a homozygous red flower plant is crossed with a homozygous white flower plant, all F1 offspring produce flower with both red and white patches, as shown in this diagram. This is because both alleles are equally and independently expressed in the heterozygous phenotype. So when F1 plants are self-pollinated, the F2 generation show the flower phenotypic ratio of 1 part red flower, which is homozygous, 2 parts of the red and white spotted, which is heterozygous, and 1 part of white flower, which is homozygous. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson. Have a great day.